on to the methods lecture. Okay, so for the methods lecture, we're going to talk first just about preparing the neural data. We won't go into like super specifics because that is going to depend on what the data is that you all are analyzing, and I think you all know best what the typical preprocessing should be for your field. But we'll talk about just like how to get it ready for a linear encoding model at least. We're going to spend a lot of time on building the design matrix because that's probably the hardest thing to wrap our heads around. And then like we've already covered basics of regression. We kind of flipped the order. But we'll kind of get into like the fitting and the cross-validation and stuff. And I'd like to get you guys onto the notebooks as soon as possible. So we'll spend most of our time on the building of the design matrix. So just a brief reminder of our goal. We want to identify how variables of interest modulate neural activity. So this could just be like we want to feed in raw video into our model, or like maybe if this video comes up, we'll want to feed in like some annotated labels. So you could use like deep lab cut to say where is the nose, and I want to feed in like how much the nose is moving as an input to my model. That could be another input. We also have auditory inputs, like in this case clicks for our task, or you could have some sort of visual stimulus. That's all what we call X, our design matrix, and we want to predict Y. So we relate it with this set of beta weights here. We already learned how to solve those beta weights, so now let's talk about how we build X and Y. Okay, so first you need to decide on the alignment scheme for your data, because that's going to determine everything, like how you structure your design matrix. So do any of the necessary smoothing, binning, whatever is typical in your field. And then the next question is, do I trialize the data or not? And so this is going to depend a bit on how you collected the data. If you collect one continuous stream of neural data, you can just leave it like that and then align all of your regressors of interest to that continuous stream of neural data. But if not, like if you're stopping and starting your recordings, you're going to need to grab a certain amount of time from each trial that's like aligned to an event so you can know where in time you are. And then ultimately, as we talked about a bit before, the shape of the matrix Y is going to be n samples, which is frames, by the number of neurons, voxels, dimensions, whatever it is you're trying to predict. And that's about it for kind of getting your matrix of neural data ready. So now for the design matrix, this is, I would say, the most time-consuming part of building these models when like actually implementing it with your own code. It's very specific to your data set and your task, so think very carefully about how you're doing that, and we'll talk about important considerations when building a design matrix. And then lastly, this is going to have a bigger impact on your results and your interpretation than like any sort of variations in fitting procedure. So if you're changing your design matrix, that's going to change your results a lot more than like, you know, selecting a slightly different alpha or like different, like I've tried a bunch of different methods for picking a best alpha. None of them change anything compared to the changes you see when you like redesign your design matrix. Okay, so when actually building the design matrix, there are multiple types of regressors that we could possibly include. So one would be what we call like an analog regressor. And I'll start with this because it's kind of the most intuitive example. Like we just have our continuous time series of neural data, and then we might have some other continuous time series, such as the size of the pupil, and we just want to relate those to each other. So that's pretty easy. We're ultimately just going to fit one weight that relates this time series to the second time series. And so yeah, like pupil diameter, the position or the motion energy of a DLC label would be something like that. Where it gets a little trickier is when we have what we would call binary regressors. So these are things where there's like only one value per trial. And the tricky thing is our model, like our samples are time. So we need to figure out a way to go from trials to time with our regressors. So we'll talk a bit how to do that. We'll talk a bit about how to do that. So yeah, that would be something like, well, what choice did the animal make on this trial? Was it left or right? And how do I code that into the matrix? And then finally, the third type of regressor, which is like sort of an extension of the second one here, would be things like trial events. So these are things that occur at specific times over the duration of a trial. And it could be aligned, or it could be happening at totally random times in the trial. So like one thing you could do is like say we're looking at the pupil or we're looking at the eye, and I want to see, okay, well, how does blinking affect neural activity? I'll basically have this sort of binary trace through time. Every time the mouse blinks, there's a 1. But how do I expand that out and see how blinking impacts neural activity both before and after the blinking actually occurred? Is that making sense? Okay. All right, so let's talk about analog regressors first. Like I said, these are pretty straightforward. You'll just take whatever your metric is. So in this case, we're taking the motion energy of some body parts. And we'll just do the same thing we did with the neural data. We're going to resample them because we need, like these cameras are probably not sampling at the same rate as your fMRI is, or your wide field, or your mesoscope, whatever. So we need to resample those and then align them to however we aligned our neural data. So in this case, let's just say we align to stimulus onset. So we grab X number of frames before the stimulus onset, X number or Y number of frames after the stimulus onset, and then we reshape those so that it's now just one long stream of time, like we did with our neural data. Binary regressors, it's going to get a little more interesting, so I'm sorry for this next graphic, but what we do is we take this vector. So let's say for any given trial, a 0 represents a left choice from our mouse, and a 1 will represent a right choice. So the way we're going to expand this out into time is actually with a series of basically identity matrices. And the way we do this is we want to say, okay, I want to know how the choice affected neural activity on every single frame of the trial. Because it's possible, like the mouse reports the choice at a particular time, but this decision in the mouse's brain is forming before that actual choice is reported. So I want to be able to know, how did choice at an early point in the trial affect neural activity? And so the way that we do that is we actually set up a number of columns or regressors for choice. So in this case, what we've done is we've set up 11 different regressors for choice. And so this is kind of part of the choice portion of our design matrix, where we go down by frames, and then across are the number of regressors. So what we do is, if there was a leftward choice, we just leave that all as zeros. It's just an empty matrix here. But then when the mouse will choose right, what we do is we add this series of ones. And I'll try to give some intuition. So this is like actual time through our neural data. This is what we would call like trial time. So this regressor right here is on the first frame of any given trial, how did choice affect neural activity? And then on the second frame of any given trial, how did choice affect neural activity? So on and so forth until we get to the end of trial time. Is that making sense for people? Okay, good. And then this is just showing the full equation again. So the design matrix will be of dimensions frames by predictors. And then our Y is going to be frames by like components or neurons or whatever. So therefore, our beta weight matrix is going to be, the dimensions will be, sorry, I'm losing my train of thought. It'll be the number of predictors by the number of things we are trying to predict. Okay.
Sorry, quick question.
Yep.
How do you decide for the OR? How do you decide how many columns I want to assign for this choice regressor?
So
Here you mentioned 11. How did this?
yeah, so I just, in this sort of pretend scheme, I'm just pretending that like with my neural data, every trial is 11 frames long. And for these kinds of binary, like whole trial regressors, what I chose to do, or usually choose to do, is just say, I want to let them explain activity across the entire trial. So the reason for choosing 11 is it's arbitrary. Yeah.
Is there, so if you have some variability
Mm-hmm.
in your kind of neural response to a punctate event, like choice,
Yeah.
um,
Variability.
temporal variability to whatever.
Okay.
You know, so let's say you make your choice, and on some trials, maybe because of the type of choice you made, the relevant neural activity is 500 milliseconds prior to your choice, and in other trials, because of that type of choice, it's 200 milliseconds.
Yeah. So.
Where, could specifying like the dimensions of this matrix help or hurt you, kind of like uncouple? If you had another variable that told you what kind of trial type, but maybe that's late, maybe you don't know what it is, and you just see a whole bunch of smear. If you had an event happening in column 2 for half your trials and column 4 for half your trials, I know you look at those independent regressors, would you kind of get like half an effect size versus if I had a much coarser framing with just say 3 columns, where I said everything pre-choice, everything post-choice, I would compact that into a single regressor. Do you see what I mean? Like the granularity
Oh, I see. Like
of this.
so do you downsample across time, sort of?
Downsample, kind of like how you're coming up with this 11 columns,
Yeah.
3 columns versus as a way to kind of compensate for
That's a.
neural smearing. I'm calling it smearing, but like
Yeah, no, I see what you're saying. That's a good question. I haven't done any of that, so I can't say how it actually influences the activity. But it is tricky because we have no idea when the choice happened, right? It happened at some point, but all of this is just aligned to the stimulus onset. So yeah, it's definitely a problem, and that might be one way of kind of overcoming. What I will talk about in a bit is like the animal, like in a different task, we have the mice lick a spout. So we have this sort of like choice regressor, but then we also have like a licking regressor, and that's time locked to the times that the lick happens. So is that the choice? Is that not the choice? Maybe not, but at least now we can align it to something, and we can look back X number of frames before the lick happened. Okay, and so that kind of leads me into this third point, which is trial events. So like the mouse can lick, and they can lick at any random time during a trial. So how do we deal with something like that? And it turns out that that's pretty similar to the binary regressors that we just covered, but you'll actually have to shift them forward or backwards in time, depending on when they occurred. And so the other thing that you need to do for these types of trial events is you define like a pre-time and a post-time. And what those specify is how long before the event occurred do I want to be able to explain neural activity with it? And then post-time, how far after the event occurrence do I want to be able to explain neural activity? So the sort of obvious case is like for a stimulus, the pre-time for that should be a 0. Because if the stimulus hasn't come on yet, there shouldn't be any way that it can be modifying neural activity. So we'll define like a time of 0 for a pre-time of 0 for stimulus, and maybe 1 to 2 seconds for a post-time. Things like motor signals, we think there's generally like preparatory activity happening before the execution of a motor command. So we do allow those regressors to explain variance before something actually occurs. Okay, so we'll go back to this sort of same example. And now I'm kind of adding in the alignment that we did. So like I said at the beginning, we align these to stimulus onset. So you can see within each of these trials in time, the 4th frame is always going to be the stimulus onset frame. And then in order to decide where you put your regressor, like let's say we have a stimulus onset, well, we can't use stimulus onset because that's what we align to. But let's say we have like a licking regressor. If it occurred 4 frames after the stimulus onset, then you'll have to shift it down, like shift that column of 1s down by 4 frames. Likewise, if a lick occurred 1 second before the stimulus regressor, or the stimulus onset, you shift it up by 1 frame. And so that's how we kind of code these variables that are not happening with any sort of trial alignment. Okay. And so finally, you assemble your analog, your binary, and your event regressors into one large matrix, and you get something like this. So this is about 8 trials of data. So this is way longer at the bottom. But you can see here, like, each of these is one trial, and we're kind of marching through time here. And then along the second axis is the regressor axis. So like you can see here, a lot of these things are like task variables. So like I think this one is the choice. This one is like rewarded versus unrewarded. These ones here that we're seeing are licks. That's why there's multiple different diagonals, because you can have multiple licks for any given trial. And then if we move over on the right side here, these are more of like our analog regressors. So these would be like, I think this one's pupil diameter right here. These would be just the facial video, and then the facial video, like the motion energy of the facial video. So that's if you actually kind of visualize the design matrix, that's what it's going to look like. Yeah, and then that's basically it for design matrix building. I think at this point we'll probably, let's see. Yeah, I think we'll move on to the notebooks, because that's really the most complicated part. And that's going to be what's most specific to your data set. So we've built the design matrix for you for this particular notebook, but now we're going to actually get in and fit a linear encoding model to this miniscope data. So let's see here. So yeah, we're just going to load the data. We're going to get some of our dependencies again. We're going to plot some footprints of the neurons and just see what they look like. And then just to kind of walk you through some of the variables that you're going to be loading at the beginning of the notebook, the design matrix it's going to be just like what I showed you. We're going to have Y raw fluorescence, which is the calcium activity from these neurons. Footprints, again, it's just so we can see what the neurons look like in 2D space. And then frames per trial is that number of frames that we chose to include in any given trial. So like in this example, the frames per trial is going to be 11, because like we marched through time, we hit 11 samples, and now we're on to the next trial. Okay, and then, yeah, I'll skip over that. And then, let's see. We'll use scikit-learn to do a lot of the fitting now. So I'm just going to go through a little bit of the documentation, so we can kind of get an idea of how we're going to call some of these things. So sklearn has this ridge CV object, which is really nice because we can actually use it to try out a bunch of different alphas, and it's just going to tell us what's the best one we should use for our model. So when we call this this object here, we have to pass an array of the alphas that we want to try. We also have to pass whether or not we want to fit an intercept, and we're going to be mean scoring our data, or mean centering our data, so we shouldn't actually need to do that. We're going to specify how we want to score it, how we want to cross-validate it. And then also there's this alpha per target parameter. And what alpha per target means is, do we want all of the neurons to have the same alpha value, or do we want them all to have different alpha values? And there are cases in which both can be right. There's one that usually leads to better performance than the other. So I'll let you guys try both and figure out what that is. And then let's see, what other stuff is there to cover? Okay, the other thing I definitely want to touch on is this pipeline, sklearn pipeline.
So can I ask about that alphas
Yep. Alphas per target?
Yeah. How does that work?
How do they implement it? Or
Not the specifics, but I guess my sense would be I'm running this regression. My alpha is telling me what my kind of penalization term is broadly across all of my betas. My betas are predicting my neural.
they are, but you have a separate set of betas for each neuron you're trying to predict. So each set of those betas can be differently penalized. Yeah. Okay.
They're not getting pooled into
Yes, exactly. Yeah. Okay, so we're going to also make use of this pipeline within scikit-learn. Pipelines are just a really nice tool to know generally because they can take your data and somewhat like a neural network, they can pass it through like a variety of different steps and assemble it into one large model. So like one thing we'll be doing with the pipeline is we're going to actually be applying all of our normalization and whatnot in the first step of the pipeline. And then in the second step of the pipeline, we'll be doing the ridge fitting. And it's generally best practice actually to do your pre-processing within folds. Because if you like pre-process your data and then split it, you can sort of run into data leakage issues where you may get better cross-validated performance than you actually should, because your testing set has some information about your training set when you pre-process them together. So we'll use a pipeline to like take care of that for us and make it really easy. It's automatically going to z-score stuff within individual folds. And then, yeah, so I think you all will definitely want to take a look at the documentation when you're learning how to call these functions. But these things are all super helpful, and they're going to work for like many other kinds of models that you want to build, not just an encoding model. Finally, there's this k-fold object, and this is just scikit-learn's way of like defining how you want to do your k-fold cross-validation. So there's a couple different parameters that we'll be passing to that. Okay. Yeah, and what we're going to be doing in the rest of the notebook is we're going to look at kind of how much overall variance we can explain for each neuron. We'll plot some of those beta weights for the neurons through time for like choice and for previous choice. And then what we'll do is, as a sanity check, we're just going to plot peri-event time histograms. So if I have a beta weight kernel that looks like, oh, whenever there's a right choice, this neuron ramps its activity way up. We should be able to see that in the PETH too. If we can't, then we know that our model is not doing something totally correct. So we'll just plot those at the end as a sanity check. And then in the third notebook, it's just going to kind of go through some more data visualizations, how we would compute that delta R squared metric that we talked a bit about in the first lecture. So I think that's mostly it for the methods at this point. I'll let you guys kind of run with these two notebooks, and then we'll be coming around to answer any questions that you might have.